Good evening, dear delegates and participants. Sorry for the inconvenience. I am Mr. Lakshmindra Sahu, Assistant Professor, Roland Institute of Pharmaceutical Sciences, Berhampur. Welcome you all to the third scientific session of Day 3, EFDP on Artificial Intelligence on Drug Development, Recent Trends and Future Prospective, sponsored by AICTE-ATAL Academy, New Delhi, and organized by Roland Institute of Pharmaceutical Sciences, Berhampur. Dear delegates, now we have with us our most respected eminent resource person of the session, Dr. Surajit Devnath sir from Women's Polytechnic AICT Tripura. Before starting his eminent talk, it's my privilege to introduce him to all of you. Dr. Surajit Devnath sir, currently working as Head Department of Medical Laboratory Technology, Women's Polytechnic Tripura, with more than 13 years of experience in biomedical research and high-level teaching. Sir participated in various academic work in institutions of Germany, Denmark, and Hong Kong, besides in premier institute lab such as AIMS, Cytogenetics, IIT Kharagpur Biotechnology, IIT Guwahati Biomedical Engineering, NB PGR Plant Genomics. He is the recipient of Young Scientist Award from different agencies such as DBT, DST, and CSIR, and recipient of Tata Trust Grant for CME course in Royal College of Pathologists, London. He is a reviewer of several Scopus ACI Index journal published by Elsevier, Springer, Biomedical Central. Along with regular academic activity, he is also engaged with several biotechnology companies in northern and western India as a consultant. Sir has conducted various workshops and talks and acted as a senior chair in international conference sponsored by national and international academic agency. Currently, Sir guiding a group of international students for their internship from Russia, Turkey, and West African countries. In the year 2020-21, Sir has filed 2 patents in India on biotechnology. With this brief introduction, I would like to request Dr. Surajit Devnath sir to start his scientific talk on fabrication of nanoscale entities and peptide scaffold for drug delivery, cutting the project cost using machine learning approaches. Welcome you, sir.
Your kind introduction. So I shall start my session now. Sir, coordinator sir, I'll request you to start the PPT while I'll explain the things.
Okay, sir. Okay, sir. Just.
Okay. So when we are talking about the fabrication of nanoscale entities and peptide scaffolds for drug delivery, so we are essentially talking about peptide-based scaffolds which have multiple different usage. So till now in biomedical research, the scaffolds are useful for generation of artificial organs, for example, cartilages, then various bone structures. Then subsequently it has moved into soft tissue as well. So today it is possible to engineer so soft tissues like pancreas as well as what do you call as liver or hepatocyte, a part of a fully functional hepatocyte build up as well as a tissue matrix. So there are multiple different usage, but when we are talking about peptide scaffolds, if it is an inert scaffold, these possibilities are highly there. But when we are talking about a peptide scaffolds, and if the scaffolding is within nanoscale range, so there are some added advantage. The advantage is the whole system acquires a whole new dimension, and several other bioactivities are integrated into it. So if we go to the next slide. Sir. Okay, so if you can see here, the nanoscale entities, there is a particular kind of interest throughout the world. What happens is a simple system. If it is in a nanoscale, so it acquires a completely new physical, chemical, and if we say physico-chemical and as well as biological properties due to its size, which is within 100 nanometer range. So, if the peptide and the peptide scaffold, which is also within nanoscale range, so the potential increases. First of all, the scaffold is nothing but a matrix. In that matrix, you can germinate specific cells, and then by using a specific growth factors, you can germinate those cells. So, what are the possibilities? The possibilities are huge. For example, today the best practices in tissue engineering are mostly using proteins such as sericin and silk fibroin and things like that. Now these proteins are functional, but somehow these are inert. So it makes a inert matrix, and upon which you can germinate cells like osteoclast and osteocytes, which will eventually become a part of the bone structure or cartilage structure. If you are growing chondrocytes on the tissue scaffold or scaffolds, so you can germinate or you can produce artificial cartilage. But what will happen if the scaffold itself is so small that it is within nanoscale range? In nanoscale range, cells will grow, but it will there are possibility that it will end up with making some kind of nano robots, which have completely new dimension for drug delivery and things like that. So these are mainly experimental setup, but in our lab we have achieved some part of it, and we are looking for collaborators where we can ultimately reach to the anticipated goal. So here today I will be discussing a part of our journey that we have already made, and how computational simulation has helped us to reduce false positives. So this is also very interesting and important when we are talking about computer aided drug design, delivery and things like that. What is the main objective? The main objective is to reduce time and reduce cost. Now how do we do it? For example, I need to make 10 different wet lab experiments, in vivo experiments, where I will find out which kind of protein entity is good for making a scaffold. Now, if I can make some assumptions through computational simulations, what will happen is my false positive, false negative or false positive, whatever you say, will be reduced to a significant extent. Instead of 10 different experimental setup, I'll end up with 5 different experimental setup, and therefore I'll reduce the cost of my lab, and I'll end up with using much less costly substrates and costly reagents. As you know, the growth factors and all other things are very costly. So this is one of our objective. So going to the second slide, sir. So there are different approaches to generate nanoscale entities. So usually we call it like okay. So usually we call it as bottom up approach or top down approach. Now in bottom up approach, what happens is we are having the small subunits, and by joining these subunits we end up with something which is bigger, not bigger than the nano entity. Okay. Now one more approach is there. In that approach, we are having a large entity, and by cutting it into small pieces, we reach the nano level. So now the bottom up approach, something is very common in both the approach, bottom up and bottom down. Both the approaches are very common in natural living cells. So I'll give you one example. For example, when during cell division the chromosomes are moving from one pole to another, so there is a thing called spindle fiber. Now spindle fibers are nano entities. Now how do they make up? These are make up with all of us already know that tubulin subunits are there, which will build up one upon another, and the spindle fiber will be formed. Now when it is building up, the spindle fiber will move forward If you can visualize by yourself, you can understand. So what will happen? The subunits are called tubulins. Now tubulins are such that one after another it can go like a garland. आप लोग जब मतलब when we give a mala to a god, then what we do? We take one by one flower and put it as a garland to take it to any of our god or somebody who we worship. So what happens is during that part also we are doing what? We are doing a bottom up approach, isn't it? So similarly, tubulin monomers are there which bottom up and makes the spindle fiber to move forward. Now what will happen when we are moving it backward? In backward, depolymerization takes place and subsequently it becomes smaller and the backward movement is there. So similar approach is also there in nano engineering. So, but in our case, we have used this bottom-up approach where we were having the single entity and subsequently we made a amalgamation or shall I say assembly, and subsequently we end up with a peptide bed scaffold. So, we will discuss it with some more detail. So, sir, going to the next slide. Yes, so these are the possibilities of nano engineering where peptide bed scaffold and other things are there. So first of all, the usage are in diagnostics by making several entities for bioimaging, where we are essentially talking about antibody based what do you call is fluorescent molecules which will be used as this thing. And then when we are talking about biosensors, here we are essentially talking about some redox receptors. That means oxidation reduction taking place, and therefore there is a change in the potential difference, and subsequently we will understand if there is an electrode. So biosensors are also a very important aspect. Then there is a therapy. Now in therapy, these most common therapies where nanoengineering is helpful is regenerative medicine, cancer treatment, and then antibacterial agents or antimicrobial agents, if we say. Subsequently, there are drug delivery mechanisms, which is a part of your curriculum. Since I am not typically a pharmaceutical scientist, this part is not very distinct for me. However, the drug delivery mechanisms are here we are interested in gene delivery as well as medication delivery. Now, medication delivery is also very important. For example, if you are making a liposome based drug delivery, so in that case, what will happen? We will embed the drug into a liposome, and the liposome will be taken by means of a mist. Something like the spray. When we will put it a spray through the nose, the mist or the shall I say the mixture of or in the or an colloid of air and water interface will enter into your nose, and the drug will be delivered. So these kind of things are already there in the market. For example, those patients who are suffering from osteoarthritis, they are given vitamin D3 through this kind of nasal route, and it acts very well because it is so biocompatible that it absorbs very rapidly instead of a capsule where you are giving a calcium in the calcium with some other excipients and then put a drop of vitamin D3 which undergoes denaturation rapidly, and therefore the ultimate this thing is very low. Availability is very low. So there are these kinds of things where we can enhance the bioavailability by using nanoscale entities. So one more very important thing that I am interested in, and I must talk about it, and this is the thing of theranostics. Now theranostic is nothing but the combination of therapeutics as well as diagnostics. So what happens is, in a single entity you can do the imaging, at the same time it can also deliver a drug. So this is also very interesting and rapidly growing thing in today's biomedical research scenario. So sir, going to the next next slide. Yeah, these are the things that what happens is in bioactive scaffolding, what happens is two or more things comes together. For example, we were interested in anti-microbial potential. So that is also embedded in a nanoscale entity. So what is happening? You are doing a drug delivery through a vehicle who is already anti-pathogenic. So what happens is there is no scope for any other pathogenic entry or things like that. So what happens is you get something which is with a value addition. So pharmaceutical industry is very much interested in value addition. So value addition is definitely there. Uh, Sir, is there a difficulty? I am not able to see the screen properly. Okay. So here these are the possibilities again of peptide-based nanoengineering. Now this photograph was taken from Cavendish Lab, Harvard University. Sorry, Cambridge University, where I have personally spoken to this thing author and taken his permission for my presentations. So what happens is here you can see what are the possibilities. So peptide based scaffolding can be used as a 3D scaffold, as I have said, a matrix on which your tissue will grow, because all tissues are nothing but a combination of cells which are growing on a matrix. Then subsequently we can have some other things, for example biomineralization and hybrid material, where you are combining fullerene or carbon nanotubes with the peptides. So there are multiple possibilities. One more is, as I have said, liposome mediated things, where you are embedding liposomes as well as peptide assemblies, and subsequently you are developing new kinds of for example, you may have heard about a COVID-19 vaccine, which is an mRNA vaccine, which will be delivered through a liposome. And the mRNA will be embedded and it will start producing protein through proper translation pathway, and your vaccine will be delivered, which will be produced in your body only. So there are excellent possibilities there. Then there are, as I have said, the fibrils. The example I have already given about spindle fiber. So these are the possibilities where nanoengineering and peptide-based nanoengineering can be helpful. So here one more thing that I would like to this thing, as you have already seen. Sir, if you click here, one more this thing will appear. Sir, can you click here? Now this is the same lab which have proposed about a nanopeptide-based vaccine against coronavirus back in 2012. So back in 2012, this lab understood that there must be some kind of this thing potential danger with the coronavirus. At that point of time, we did not call it coronavirus 2 because it was all coronavirus 1. So if you see the name in the paper, they have written as about coronavirus. They have not written about COVID-19 or things like that, because it was not there. Eventually, it has modified itself, and it has become a pandemic today, and all of us already know that the thing is that, like, if you take a vaccine also, you are not spared, and every day new strains are being developed, and it may happen that for every new strain you need to develop a vaccine regularly. So these are all possibilities which also focus to our next advantage of nanoengineering, which is personalized medicine. So I would also like to this thing, sir. Thank you for the next slide. So here what is happening is, usually a single therapy does not fit all. So for somebody it is very good, for somebody it is not effective at all. So what does it mean? It means that there is a desperate need of personalized medicine. So for example, somebody is affected with the alpha strain of COVID. So if he is taking Covaxin or Covishield, it may happen that he have developed no immunity at all against this alpha or lambda strain. So what he will do? He will develop symptoms and eventually he may die. So what happens is, you need to develop strategy in order to tackle all this. So one such strategy is personalized medicine. So to give a proper therapy the way person needs, it will not be a single size fits all, and like this it will be a possibility in the future. In near next 50 years, it will be it will be a case that every new this thing requires a different kind of therapy altogether. So nano engineering can help desperately in this regard as well. Sir, go to the next slide. Yeah, this is the yesterday's case, and tomorrow's case will be the bottom case, that is personalized medicine. This is what we wanted to speak. So sir, go to the next slide. Okay, now these are the possible things that can be done through tissue engineering and nano engineering. So nanoscale entity can be entered in the body through various modes. As you can see here, the first mode is phagocytosis. What happens is the drug that you have embedded in a nanosphere will be entered into your system through a phagocytic vesicle. What will happen? It will be delivered somewhere where your macrophages are there. The macrophages are antigen presenting cells which are having phagocytic property. They will take up the nano drug and they will deliver it to a particular site, and eventually your medicine will be delivered. So the other possibility is what do you call is receptor mediated endocytosis, very important concept, where clathrin coated pits and all things we know, which is applicable for those drugs which are lipophilic in nature. So if you have a lipophilic liposome mediated delivery, so what can happen is a specific endocytic vesicles will be formed and eventually it will be delivered. So this is how this thing can be there. So the next slide, if you see, it is again the possibility about what nano engineering and nano scaffolding can do. A very simple thing is, for example, if you are embedding some enzymes, which is very common in pharmaceutical industry, you are immobilizing an enzyme and increasing its shelf life. For example, let me give you one specific example of capsule based or capsule based enzymes that are delivered. There are very good industries, companies, pharmaceutical companies. For example, Abbott is a forerunner in this kind of work, where they have embedded lipase, protease, and amylase in a matrix in so stable way that when you take this medicine, then only they will start their function. And the brand name is Creon, and it is very famous and it's a life-saving drug. So there are possibilities to embed enzymes in a matrix, and not only enzymes, therapeutic proteins on a matrix, and thereby increasing its shelf life as well as sustained delivery process, which is an important thing in this thing what do you call is pharmaceutical industry, though I am not directly this thing there. So here also it can directly projected to a cancer or a metastasis, and which will do wonders. We have had excellent clinical trials and lab scale studies where nanoscale entities have given excellent result. Then there are possibility to develop 3D organs, or by 3D printing we can one day print organs and then do the what do you call is transplant without any problem of donor receptor incompatibility and so many other things. So these are all the possibilities, and the work has been cited from a particular work where we have taken proper permission to cite this with the figure. Then sir, if we go to the next slide. So there was a video. I am very much sorry that I cannot able to play it, but if the coordinator wishes, we can able to do it in a different way. So sir, coordinator sir, is it possible that you go to YouTube and then write the keywords that I'll say, and after that one video will come and you play the video in a to YouTube, then search for
Tell me, sir.
Okay, yes. So sir, if you go to YouTube, then we can write nanoscale delivery, Monash University. Or nanomedicine, sir, write nanomedicine and then make a space and then write Monash University, MONASH. So I hope it can, yes, nanomedicine, then space Monash University. If it is there, MONASH, Monash, MONASH University.
Okay, sir.
If you can share the screen, I am sorry that I am not able to do it in the way I anticipated. Sir, if you share the screen, I can tell you which exact video is there. Sometime it may happen that you cannot able to find it. So many other videos will come.
Okay,
So participants, yes, participants will also excuse me.
Welcome to era of nanomedicine. That one, sir.
Sir, is it possible to share the screen, sir, so that I can pinpoint exact thing?
Okay. Just a second, sir.
Sir, participants are there, no, sir, in this session?
Yes, all are there, sir.
Okay. I hope that the specific video we can find. I also forgot. No problem, sir. No problem.
Now it is
no,
visible, sir.
Welcome to no, sir. These are sir down, sir. Some more video.
No, it is visible to you, video.
It is visible to me, sir. But the specific video is not there. Sir, if you
So
go
I'm
to a
going-
little down.
I'm
Please
going
go, sir.
down.
Please go, sir. Let me see the video once. Not there, sir. 1 second, sir. Let's see what is the starting. No, it is not there. These are all relevant, but I don't want to give the other thing, and I want the exact one only. 1 second, sir.
No, I have typed here nanomedicine.
Yes. You have typed the correct thing only. 1 second, sir. I'll find out.
Yes, you can check.
Sir, you write nanoparticle based drug delivery, sir. Nanoparticle based drug delivery. Then you write Monash University, sir. I am sorry, sir. Nanoparticle based drug delivery, and then Monash University. Sir, now please. Yes, sir. First video, sir. Sir, you go to yes. This is the one. First one, sir. First one. Yes. Thank you. Thanks a lot.
Okay, sir.
Sir, please play the video without sound, sir. I shall speak. So here we will be specifically talking about. Now this is a fluorescent microscope. If you can see, switch off the sound, sir. Is side mein, So what happens is here only, sir. Yes. So this is a fluorescent microscope where different types of cells are seen, and if you can see, these are very highly bright. Okay. Now if we go close, so we will see. We are going close. So what we have done is we have used one fluorescent antibody. Those cells we are normal are producing pink color light, and those cells which are cancerous are producing blue color light. If you can see, this is a cancerous cell. Okay. Now what we have done is we have engineered a nano-based drug, and we are delivering it. Now what happens is whenever there is a cancer on a cell, it produces a specific antigen on the surface antigen. So the drug is such that it binds with that specific antigen only. So sir, play, sir. Sir, play the video, sir. Let the video play, sir. Now see here, the nano-based drug is going, and reaching the specific site. These are the antigens which are present on a particular cell only. Yeah. Now these are the receptors. See. Okay. Now these are getting binding. Okay. After the binding, the drug will be internalized. See. Now it is internalized, and now specific receptors will take it to the nucleus, directly to the nucleus, because it is destined for nucleus only. Okay. The clathrin coated piths are there. Now it will directly deliver to see it is directly delivering. Now this is the endoplasmic reticulum and Golgi. It is bypassing endoplasmic reticulum and directly going to the nucleus. Here you see. This is the nucleus. So after going there, what is happening? It is internalizing. Internalized into the nucleus. Now the drug will be delivered. Okay. So what happens is, usually in pharmacokinetics and dynamics, it is metabolized and then it becomes a drug. We give a prodrug, then it becomes a drug. Here also it is a prodrug. Now the drug is getting delivered. Now the drug is getting delivered. So if you can see, yes, the drug has been delivered, and now the cancerous cell only will die. Here you see, by a something called what? Necrosis, or we shall say it programmed cell death or apoptosis, whatever kind of signal you give. Thank you, sir. Thanks a lot, sir. Now go to the presentation, sir. Now this is how nanomedicine and personalized medicine will work one day. Okay, sir, go to the next slide.
Just a second, sir.
Yes, sir, no problem at all. Okay, so going to the next slide. Okay, so these are the possibilities. The same lab, they have developed cartilages, and they are trying to also engineer some drug delivery mechanism through nano-based nano entities. Sir, going to the next slide. There was this worksheet. It is difficult now for me. Sir, going to the next slide. Okay, these are very good specific examples where nano-based scaffold on which the chondrocytes has been put, and a whole new the cartilage has been developed. Excellent results by somebody else. Sir, going to the next slide. So these are the peptide based nano entities where a specific kind of structural geometry is needed in order to form a scaffold. Here it is showing that a specific and parallel beta sheets are needed on the protein, so that the assembly is there. Sir, going to the next slide. So as I have said, the bottom up and top down, both the approaches are there in the nature itself, and from nature only we have been motivated to do nano engineering. So sir, going to the next slide. So these are the possibilities that we can have. For example, one, we have a small peptide, then we make a peptide nanotube, and then we self-assemble the nanotube to become a array of peptide nanotubes, a bigger one. So this is how the whole this thing takes place, and there are different approaches through which we can do nano engineering. Sir, going to the next slide. Okay, now one more video, sir. In that video so much is there, so we shall definitely able to try it. Sir, again, sir, I'll request you go to YouTube and then write about self-assembling nanotube, self-assembling nanotube, and then write ACS, self-assembling nanotube, or just wait, sir. We shall write about, just 1 second, sir. Yes, one second, sir. self-assembling peptide. Self-assembling peptide, and then write ACS, American Society of American Chemical Society, ACS. Self-assembling peptide, then ACS.
Sir, you can see now. Sir, it is visible.
A different previous screen, sir. Okay, sir. Go to the down, sir. Self-assembling ACS. थोड़ा down, sir. अच्छा, sir. Sir, please write ACS को रहने दीजिए. नहीं, American Chemical Society नहीं, sir. Please write ACS. All big. ACS. हाँ. S sir. ACS. हाँ, अभी एक बार देखिए सर. अरे self-assembling. One second, sir. Self-assembling. Angiogenic. Sir, please write self-assembling angiogenic. हाँ. Angiogenic. ANGIO. Angiogenic. GENIC. हाँ. Coming down, sir. Coming down. दिस वन सर. सर वाई का माफिक जो है सर, थोड़ा ऊपर जाइए. एक वाई का माफिक ये वाला करिए सर. ये वाला करिए सर. ये वाला. थैंक यू सर. थैंक यू. So this has been published by an Indian author only, where he have shown how self-assembling peptide can be functionalized. Excellent results. So angiogenesis, as all of you know, is a process which which is nothing but formation of new blood vessels. So in order to make a functional peptide, what he did was he initially did an assembling, and this assembling he tagged a particular thing which is angiogenic, a particular growth factor. So eventually, when it has been reported in ACS Nano, a very famous publisher. So this is the video. Now this was the hydrophilic domain is there, and a hydrophobic domain is there. So what happens is it is something like amphipathic. Now there is an angiogenic peptide. Now here you see how the peptide assembly is going on. It is self-assembly only. You need not to put any energy, and all by itself it will make a sheet. Please see the sheet. This is the sheet. So there are two domains. In antiparallel beta sheet has been formed, and on the terminal regions there are the angiogenic peptides. So what happens is, if you, for example, somebody is growing from some heart ailment, so this whole structure will be given, and what will happen? The angiogenic drug will be delivered. Very, very good results. So this is how a large scaffold has been made, which looks like this, and the whole process will be delivered near the heart, and the angiogenesis will start. So they have got excellent results. Sir, going to the slide. So this is the possibility of nanoscience, which can bring about rapid shift. So now I shall talk about how the simulation can help. Okay, so in our lab, we identified two different peptides from different sources and identified their bioactive potential, and subsequently we simulated these proteins. I shall directly go to the results now. Our results. that we have already published. Sir, going to the next slide. Now this is a nano scaffolding. Sir, going to the next slide. Going to the next slide, sir. This is highly complex nanofibrils. So going to the next slide. This is all you know that a drug should have this kind of properties. Sir, going to the next slide. Going to the next slide, sir. So these are the properties that can have. Sir, going to the next slide. Here what is happening is they are making a peptide scaffold, but they have also have to check whether this peptide scaffold, when they will put it in an animal, it should not cause any infection. So what they are doing is they are checking its anti-infective potential also. But in our case, the peptide itself is highly antipathogenic, and therefore this potential is already there. So these scientists, what they are doing is they are adding curcumin, they are adding some kind of antibiotic to their peptide scaffold, because they need to make the peptide scaffold anti-infective. But in my case, the anti-infective property is already there. So it is an added advantage that we are trying to say. So going to the next slide. So this is our initial result that we published some 3-4 years ago in a SCI journal only. Sir, going to the next slide. So this is the simulation pipeline that we are looking forward, where the next 4 steps. Now this is a process through which one should go before developing a product. So this is the result where we have identified our bioactive peptide and then try to simulate it. Sir, going to the next slide. So this is the peptide which has been isolated in our lab, and when we made a scaffolding, we saw that the scaffolding is within nanoscale range, and at the same time we also saw that it is highly thermodynamically favorable and stable. So sir, going to the next slide. So we have done the assemble into the nanoscale dimension, and then the nanoscale dimension is also highly thermostable and energetically stable. So for example, you make a nanotube, and when you put it in blood, you see that it is getting distorted or it is getting destroyed, because blood is a highly dynamic environment. So much of ionic change, so much of pH change, so many other things. So in order to prevent all this, it should be thermodynamically stable. So this is our first peptide which is within nanoscale range. And then going to the next slide, sir. So molecular visualization is not possible now. Sir, going to the next slide. Now this is a duplex peptide. So one peptide was there which we isolated from natural sources. Then we make a scaffold of 2 peptides. Now this is the second 2 peptide assembly. So going to the next slide. Again going to the next slide, sir. Now this is the triplet. No. Before one, sir. This is the triplet where the 3 different peptides are there. Still they are making highly stable, energetically stable thing, and then they are also in nanoscale range. Sir, going to the next slide. This is the energy landscape for a single peptide. It was approximately minus 30 kilojoules, as you can see in the last column. So going to the next slide. When we make it a duplex, it became more than 100. That means stability increasing. So going to the next slide. When we made it a third triplex, it's increased significantly. So it means that the nanopeptide assembly is getting stable, and it is also within nanoscale range. We counted its dimensions through molecular visualization tools. So going to the next slide. Next slide, sir. This is again the domain motions through a Brownian molecular dynamics, where we saw that by putting the peptide assembly, stability is increasing. So going to the next slide. So there were one movie file from 5 picosecond simulations we projected and collected and made a 1-minute video. And I'm very sorry that I'm not able to show you the video. Sir, going to the next slide. This is the second peptide, similar results. 2 peptides we isolated, as I have said. So this was the second peptide, similar results. Sir, going to the next slide. 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 This is the energy landscape which shows that yeah, fine, sir. So again, here again, triplex peptides are showing highly stable energy. So please, we can remember that whenever the total free energy of an entity is reducing, that is in negative magnitude, it means that the degree of disorder is also reducing. It means that it is more stable. So negative magnitude means more stable. So here also we are seeing that there are some distortion, but overall it is stable only. So going to the next slide. Going to the next slide, sir. So while doing this, we also tried one more thing that was not able to, I cannot able to show here, that we changed the structure of the peptides. We isolated a peptide, then we done some kind of site-directed mutagenesis. Now, site-directed mutagenesis is a process by which you can change the amino acid residue. For example, in the 9-chain residue, one proline is there, and now proline is making some disturbance or some steric clashes, if we say. So we are replacing proline with a valine by using a clone based, or it's a small cloning that should be done, but the kits are available. So overall we call it site directed mutagenesis, and by doing this we are changing the some points of the peptide, and we are seeing that there is an increase of the stability. But the problem here is there is no tool to assess the stability of the peptide after mutations. So for last 4 years we were working on it, and on 2020-21 we have successfully filed the patent for it, and we are also talking to private companies to commercialize our patent when it is granted to us. So going to the next slide. These are some structural data which are there, that is the setup of the whole process and the equipment. Sir, going to the next slide. These are okay, going to the next slide, sir. These are some experimental results that we have found. Sir, going to the next slide. These are some technical specification of the process. The one more letter that we received from Government of India, and we are expecting that this thing testing will be done soon and the patent will be granted. Sir, saying so, I'll conclude my session today, and I'm thankful to the authorities that they have given me an opportunity to talk in this special session. And I'm thankful to the whole institute as a whole, and I'll be happy if I can answer some of the queries from the participants. If it is there, I'll be very happy to try to answer them.
We have received some questions, sir.
Okay, great. They have asked how we have isolated. So the isolation process requires some kind of, initially you can do solvent extraction, and then some salt extraction also, if it is good enough. Then subsequently you can do some filtration. Ultimately you have to do chromatographies. It depends which kind of protein is there. You can do HPLC, which is little bit costly. Otherwise also you can simply do size exclusion, but you cannot do SDS page, because if you do SDS page your peptide will be gone. You can do it initially in order to find out what is the character of your peptide, but in order to isolate them, if you use SDS page, it will not be very wise. It is good if you use some for example filter based methods, and then some dialysis, and then eventually if you do some chromatography, it will be good. So this is how I should answer it. Any more question? If it is there, I will be happy to answer it.
Yes, one is there, sir.
Okay. Plant extract we used to generate the, no, peptides are peptides by using what, sir? By using proteolytic enzyme. No. Plant extracts are there which can help you to make biogenic nanoparticles, which is very famous everywhere. For example, if you have a silver chloride solution of a particular molar concentration, then you add a particular concentration of your this thing phytochemical, and eventually the phytochemical, what it will do, it will eventually reduce it, because in the silver chloride solution it is in oxidized form. Once you reduce it, it becomes particle silver, and it becomes a nanoscale silver, and it has excellent activity. For example, today if you go to a shop, you will find a soap, Lifebuoy soap, which they'll say that it is having the capacity to kill bacteria because it is having silver. So silver nanoparticle they have embedded in the saponification process, and then it is a wonder thing, or something new which is not at all new for the scientific community and very old thing, but in the commercial scenario people will think that, oh my god, very hi-fi thing is there. Anyway, so this you can do, but for peptide I have no idea also, but I do not think that in peptide, because in order to make small peptides you need to have very strong lipolytic process, which plant extracts. Papain is there, but it is not a true plant extract you are trying to say. I think you are trying to say about some polyphenols and things like that, which are usually there in plant extract. That may not help, but I cannot say because I have not tested it. But you can definitely give it a try. It can be a good idea.
Yes, yes. Thank you, sir, for your nice presentation.
Okay,
And,
sir.
Thank
Okay,
you
sir.
for sharing your valuable time in your difficult situation. So
Okay,
in
sir.
future also we can collaborate any research activities
Sure, sure,
with
sure. I am
our
very much looking into
institutes.
it. Yes, yes, sure,
Thank,
sir. We are very much looking into it. Thank you.
thank
Thanks a
you.
lot, sir.
Thank,
Okay, sir.
thank you. Lakhinder sir, proceed.
Uh, Thank you, Dr. Surjit Devana, sir, for sharing your divine knowledge on fabrication of nanoscale entities and peptide scaffold for the drug delivery. Sir, you focus on nanoembolism and scaffold molding and type of peptide. And really, the video is knowledgeable. Thank you so much, sir. May I now request our respected coordinator of EADB, Dr. Bisumon Sahu, sir, to facilitate our eminent speaker with a e-certificate as a token of respect and gratitude.
Sir, on behalf of Roland Institute of Pharmaceutical Science, please accept this e-certificate as a
Yes, sir.
token of love and affection, sir. Please accept this.
Sir, sure, sure. My privilege. Thank you. Thanks a lot, sir.
Thank
My
you.
privilege, sir. Thank you.
Thank you once again, sir. Please take care,
Okay,
sir.
sir. Okay, sir.
Thank you.
Okay, sir. Okay.
Dear participants, with this we conclude officially the scientific session 3 of day 3. For tomorrow program, the link will be shared with your registered email ID and WhatsApp group. Thank you all for your active participation and kind cooperation. Thank you all.